All right, good morning. Thank you for attending my presentation about understanding the apple volatilome. My name is Tayab Soomro, and I'm a master's student in the Faculty of Agriculture at Dalhousie University. Today, I'll talk about what determines an apple's aroma and what regions in the genome distinguish the apple based on their aroma. Apples are one of the most consumed fruits in the world. In Canada, apples represented 43% of its fruit production, making it the second most valuable fruit crop in the country. And the apples that are grown within Canada, Nova Scotia is responsible for 2.5 million bushels, or about 9% of apples. But why don't we see all these different apple varieties in supermarkets? Well, that's because only about a handful of these varieties actually meet the right conditions to make it to the market. One of these conditions is the flavor of the apples. But how do we breed for better flavor? Well, we first need to understand what flavor is composed of. So as a consumer, when you bite into your favorite apple variety, a major determinant of how much you like the apple comes from its taste and aroma. While taste is largely determined by sugars and acids that constitute the 5 basic taste types, like your bitter, sweet, sour, salty, savory, the aroma is a complex trait that is determined by various volatile organic compounds that differ between different apple cultivars. Aroma is characterized as binding of these volatile compounds that are emitted by apples to the receptors in our olfactory system that is located on the back of our nasal cavity. Depending on the type, abundance, and ubiquity of compounds that bind to the receptors, we experience different aroma. Some of the types of volatile compounds we can expect to find are alcohols, aldehydes, and esters. So I like to use the analogy of bridging the gaps when explaining my research. So there are 2 major gaps. Number 1 is understanding the relationship between the volatile compounds and the underlying genetics of apples. And number 2 is the relationship between the volatile compounds and the consumer likeness. So whether the apple is liked or disliked by consumers. These are the 2 major gaps that need to be bridged before arriving at a solution of improved apple aroma. My research is focused on the first gap here. In other words, I focused on understanding the metabolic and genetic complexity of apple aroma. The insights that are generated from this study can be channeled to consumer panel studies, i.e. the second gap, to identify the key volatile compounds that A, can be altered through the genetics, which is important, and B, that are highly favored by consumers. But let's talk about how I achieved my part of the research. And before I do that, first, I think it's worthwhile to take a little bit of time to explain some of the terminologies that I'm going to be using throughout the presentation. Aroma compounds are organic compounds that produce aroma. And for the rest of my presentation, I'll try to consistently refer to them as volatile compounds. Next, I frequently use the term volatilome. This refers to a collection of all the volatile compounds of an organism. So the apples that formed the foundation of this experiment belong to the Apple Biodiversity Collection. This is an orchard located at the Agriculture Research Station in Kentville, Nova Scotia. This orchard has over 1,000 different apple cultivars with incredible diversity in terms of their appearance, harvest times, smell, and other numerous characteristics. This is like the United Nations of apples. To detect the volatile compounds that are emitted by apples, we use a technique called gas chromatography, which is coupled with mass spectrometry. This technique is commonly referred to as GCMS. So let's imagine a journey of an apple from a tree to the GCMS machines. Apples are first harvested, and after about a month of cold storage, the apples are sliced. 2 random slices are flash frozen and stored in the -80 degrees until they're ready for GCMS analysis. When these samples are ready for GCMS analysis, they're blended and they're mixed with the salt solution. After which we can subject them to GCMS analysis to quantify the volatiles. So let's get started with some of the exploration that I did with the volatilome of these apples. So the first question that we wanted to ask ourselves is, what are the most abundant compounds in our data set? What are the most ubiquitous, right? So note that the compound is abundant if it is present in high amounts in a sample. And a compound is ubiquitous if it is present across many samples. So each dot in this scatter plot represents a volatile compound, and the x-axis represents the ubiquity of each compound, or how many samples this compound is present in. So you can see that the x-axis goes up until 515, which is my sample size And so there are compounds that are present in nearly every sample. But remember that a compound can be ubiquitous or present in many samples, but not as abundant or present in high amounts, right? So in order to capture that, the y-axis represents the abundance of each compound. So the dots that are on the bottom left here are compounds which are unique to only a few apple cultivars, and they're not very abundant. That means they're not present in high amounts. We can contrast these to the dots that are on the top right. They're found in almost every apple and are present in extremely high amounts. So what are the major takeaways from this plot? The main thing to notice here is that butyl acetate, which is mentioned here, shown here, is two times as abundant as the next abundant compound, which is hexyl acetate. And in fact, the handful of compounds that I actually have labeled here account for almost 98% of the total volatile abundance. We then assigned each of these compounds into different categories. So in our data set, we have 13 different volatile compound categories, such as aldehydes, esters, alcohols, etc. So in the first bar plot here, I've grouped the compounds, I've grouped the compounds to the categories, and I've counted up the number of volatiles that belong to each category. That's the first bar plot here. And in the second bar plot, I summed up the abundance of each volatile category across all samples. So for example, if I wanted to get the abundance, total abundance of alcohol, I took the values, abundance values of all the volatile compounds that belong to alcohols across all samples, and then I summed up their abundance values. So generally, the major takeaway from this plot is that we find esters, aldehydes, and alcohols as being the most abundant and ubiquitous categories of compounds, as these two plots show. And this is also consistent with what is observed in other crops such as tomatoes, melons, and pears. Next, we wanted to explore the apple accessions, how the apple accessions are related to each other in terms of their volatile profile. So in order to do that, we used what's called principal component analysis. In simple terms, PCA reduces the dimensionality of large data set into principal components that explain the most variance. So in here, these components are ordered by highest variance to lowest. So PC1 explains the most variance in our data set. PC2 explains the second most variance in our data set. So plotting these two against each other gives us a picture of relatedness among all the samples in terms of their volatile profile. In the biplot, we have PC1 on the x-axis, PC2 on y-axis, and each dot is an apple cultivar. And we can see that some apples are close together, whereas some other apples are further apart in terms of their volatile profile. You'll also notice that I've colored the dots by harvest date. This is because we find a correlation between PC1 and harvest date. And in fact, harvest date is significantly correlated with PC1 with an R-squared value of 0.17. That means 17% of the variance in harvest date is explained by PC1. So if we were to draw conclusions from this suggests that the time it takes for apple to ripen is associated with its volatile profile. So, as mentioned before, time it takes for apple to ripen is associated with its volatile profile. But how does the volatile differ exactly between early and late harvested varieties, right? So to answer this question, I have on x-axis the harvest season. This is the harvest date based on Julian days, which is the days that have elapsed since the start of the year. And on the y-axis, we have the number of volatiles detected. We see that the harvest date varies over a 3-month period from late August to November. I'm running out of time here, so I'll quickly wrap up here. But the major takeaway from this plot and this other plot, which is the total volatile abundance, is that early harvested apples generally have higher number of unique volatile compounds, as well as higher abundance of volatile compounds. And the reason for this is that when you put an apple in 1 month of cold storage, the apples that are different, that are early harvested versus late harvested, go through that 1 month storage differently, and they use their metabolic processes differently. Whereas some are fast tracked, which is the early harvested, and the late harvested are slow tracked. So that's why we see these differences in the volatile profiles. I don't have time to go through the genetics, so I'm so sorry I didn't organize this well. But the major takeaway is that we find a major hit at the NAC locus, which is the ripening gene, which I mentioned before about the ripening process, that controls the ripening of apples and other numerous crops. So the most important message here is that apple ripening gene seems to be regulating not only the ripening process, but also two of the major most abundant and ubiquitous compounds, which are 1-hexanol and 1-butanol. So the conclusions are that volatile compounds are used to assess the aroma, and understanding the genetic control of aroma is important to breeding to improving the apple flavor, and which is what I have attempted to do here. And that's pretty much it. Thank you very much for attending.